Hi, let's discuss cell cycle regulation. This is an important topic for CSIR examination, and the type of question asked are always complicated. So, what you have to do, study in a well-planned manner that help you solving questions from this area. What is cell cycle? Here you can see cell cycle is divided into four major phases. What are they? G1, S, G2 and M. G1 is the preparatory phase. We can call it as first gap period. And next is S phase, that is synthesis phase where DNA synthesis occur. And next phase is G2 phase. It is known as second gap period. After progressing through the G2 phase, cells begin mitosis, that is M phase. And there are three transition points in cell cycle regulation. The first point is known as start. It occurs at late G1, and this point determines whether the cells can pass through G1 to S. And for passing through this transition point, cells requires activation of certain special factors. And next transition point is G2M. Here also the cells need special factors that we can study later. And third transition point at the middle of mitosis, and it determines whether the cells completed cell division and re-enter G1 of next cycle. What is the difference between interphase cells and mitotic cells? In interphase cells, the nuclear envelope is intact and the chromosomes are not condensed. Whereas in mitotic cells, the nuclear envelope is absent and replicated chromosomes are highly condensed. When interphase cells in the G1, S or G2 phase fuse with the cells in mitosis, what happens? Their nuclear envelopes retract and chromosome condensation. The interphase cells nuclear envelope retract, their chromosome condensation occurs. Let's check these ones. If G1 cells fuse with mitotic cell, what happens? Chromosome condensation. If S cells, that is cells in S phase, fuses with mitotic cell, here also chromosome condensation occurring. Next case, if G2 cells fuse with M cells, that is mitotic cells, here also chromosome condensed and doubled. Why? Replication already occurred here, that is G2. What we understood, some diffusible factors in mitotic cells can induce mitosis in an interphase cell. We know that these factors are the mitotic cyclin CDK complex. These diffusible mitotic cyclin CDK complexes cause chromosome condensation in interphase cells when they are fused with mitotic cells. Let's check this example. If G1 cells fuse with cells in S phase, what happens? DNA synthesis will begin in the G1 cells. How these experiments are performed? The fused cells are exposed to radio-labeled thymidine. The label was incorporated into the DNA of G1 nucleus as well as S phase cells. What happens if G2 cells fuses with S phase cells? G2 plus S. Here, no incorporation of label thymidine occurred in the G2 nuclei. What we understood? The diffusible factors in an S phase cell can enter the nucleus of G1 cell and stimulate DNA synthesis there. But these factors cannot induce DNA synthesis in the G2 nucleus. Now you have studied everything. Everything is clear. I hope you have understood everything. So read the following question and answer it. And write the answer in comment box. If you like this video, please subscribe and share. Thank you.